Hello everyone, my name is Rahul. In the previous video, we had discussed about an overview of cell. In this video, we are going to discuss about prokaryotic cell. Prokaryotic word is made up of two words. Pro means old and karyo means nucleus. So these are organisms which have a primitive type of a nucleus. So in prokaryotic organisms, the nucleus is ill-defined and it is known as nucleoid. So true nucleus is absent. Nuclear membrane it is also absent. So you can see that nucleoid in this diagram and the genetic material is lying freely inside the cytoplasm. There is no nuclear membrane. After nuclear membrane we have nucleolus, which is also absent in case of prokaryotes. There is no nucleolus. Nucleolus refers to a dense body which is made up of collection of ribonucleoprotein and ribosomes, which is seen inside the nucleus. But since there is no nucleus, there is also no formation of nucleolus. After nucleolus, ribonucleoprotein is also absent, although ribosomes are present and are of 70S type, where S stands for Swedberg unit. Now ribosomes, they are made up of ribonucleoproteins, but ribonucleoproteins itself are absent. That is, there is no independent presence of ribonucleoprotein, which is seen in case of eukaryotes but not in case of prokaryotes. Ribosomes are cell organelles which are known as the protein factory of the cells, and these ribosomes are of 70S type in prokaryotes. S stands for Swedberg unit. It is a unit of measurement of the rate of settlement of these ribosomes when they are present inside a centrifuge machine. So depending upon how fast or how slow these ribosomes will settle, we define them on the basis of this Swedberg unit. After ribosome, we have the presence of chromosome which consists the genetic material. It is usually circular and single. So a single circular chromosome consists of the genetic material. There is extra chromosomal DNA which is present in plasmid. These are circular extra chromosomal DNA which are freely present in the cytoplasm and can be transferred from one bacterium to another bacterium. Plasmids provide certain special characteristics to bacterial cells, such as resistance against antibiotics. So if there is a bacterium which is a plasmid which has genes for antibiotic resistance, and this plasmid is transferred to another bacterium, that bacterium will also become resistant to a particular antibiotic. Now after the presence of extrachromosomal DNA, we have cell membrane. Cell membrane is more or less similar to eukaryotes, but there are no sterols. There are no sterols except in case of mycoplasma. Except in case of mycoplasma, there are no sterols in the cell membrane, whereas in eukaryotic cells, the cell membrane has ample amount of sterols. Cell organelles Cell organelles are absent except ribosomes. So ribosomes, these are non-membrane bound organelles. They are present, but rest all the other membrane bound organelles, they are absent. The site of respiration, the site of respiration, it is known as mesosome. Mesosome, it is a extension or inward depression of the plasma membrane. Mesosome also helps in cell division. So respiration, it takes place inside the mesosome. So this plasma membrane which is going like this, it will take a dip inside the cell, and this will be known as a mesosome, and it is going to help in the process of cell division, and it is also going to help in the process of respiration. After the presence of mesosome, you can see in the diagram that the bacterial cell or the prokaryotic cell shows the presence of cell wall. So cell wall is present except in case of mycoplasma, where cell wall is absent. After the presence of cell wall, there are certain appendages which include flagellum, fimbriae, and a third appendage known as pili. So three appendages are present. One is fimbriae, which help in adhesion to a surface, that is to stick onto a surface. Second is the presence of flagella, which help in locomotion. Third is pili, which help in conjugation. Conjugation, it is a type of primitive DNA transferring mechanism. It is a primitive type of DNA transfer mechanism seen in case of prokaryotes. So fimbriae, flagella, and pili, these are the three appendages that can be seen in a prokaryotic cell. Now the prokaryotic cell sometimes also shows the presence of a tough layer of capsule, a tough layer called as capsule. It may be present or it may be absent. On the presence or absence of capsule, we have two types of prokaryotes. One are known as encapsulated and the other are known as non-capsulated. And it is very difficult to kill these encapsulated organisms because this capsule is very tough and resistant to the action of antibiotics. After the presence of capsule, we also see that there are certain Bodies, certain aggregations present inside the bacterial cell, which are known as inclusion bodies. So inclusion bodies can also be seen. These inclusion bodies usually perform the function of storage of certain substances which may be required at a later time. So with this, we finish off with our discussion on the prokaryotic cell and overview. See you in next video.